So welcome everyone. I'm Sophie Siove. I'm the CEO of the Partnership for Observation of the Global Ocean, POGO, which is the lead organization for OBON. So welcome to this fourth webinar in our series, which is introducing and providing updates on OBON endorsed projects. So the way this session will run is that I will give a brief overview of OBON for those who maybe aren't familiar with the program, and then we'll have our presentations from the OBON projects, and I'll introduce those in a little while after the presentation. So let me just share my screen. Always the tricky part on those two screens. So hopefully you can see the presentation.
Yes.
Okay.
Yes, it's
Great.
working fine.
Thank you. Okay. So as I said, I'm the CEO of POGO, and POGO is the lead organization for OBON, which is a UN Ocean Decade endorsed program. You can download our strategic plan from this link, or use the QR code to find out more about OBON's strategy. But in a nutshell, our vision is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. And OBON's mission is to create a hub for biomolecular measurement of marine life. So we have these four main objectives, which are also summarized in this next slide, which is a diagram of kind of summarizing the strategy and the objectives. So the objectives are essentially around development of networks and resources, innovation and technology development for biomolecular observations, enhancing information use through data management and models and things like digital twins, and ultimately informing policy and informing a sustainable future. Why my slides aren't moving? So OBON, in the decade structure, OBON is a program, and underneath decade programs, there are a number of projects. So in OBON, we currently have 20 Ocean Decade endorsed projects, and some of these are being presented in this webinar today. And the webinar series is in total composed of 5 webinars, which will eventually introduce almost all of the projects. So these are the OBON sampling locations, showing some quite a good global coverage, but generally OBON stations tend to be concentrated around coastal areas. So we're trying to get more representation of open ocean projects, and in particular things like repeat cruises, which we invite during every call for new project applications. We also invite cruises that are doing biomolecular observations to join us. So in terms of the governance and organization, the program is coordinated mainly by the POGO Secretariat at the moment, and so there are a number of staff in the Secretariat that support OBON, and also we currently have a part-time program manager, Luz, who's helping out with organising the programme. We have scientific advisory committee of around 20 people, which is chaired by Margaret Leinen, and a smaller executive committee that meets more regularly to keep the programme on track. We're in the process of setting up a user advisory committee, hoping to get that up and running before the end of the calendar year. And we also have a number of working groups and some, like the policy one, that's still currently being planned. I'm hoping to also get that started before the end of the year. So OBON has run a number of workshops, and we're particularly focusing on bringing in new regions into OBON's work and getting new collaborators from around the world. So we had a very successful workshop at the Westpac, IOC Westpac Symposium earlier this year, where we had an OBON eDNA workshop, which brought in around 80 participants and helped us to raise awareness about OBON and how people in the region could get involved. We're currently planning an eDNA for fisheries management workshop for spring of next year, and a bit later on a network and capacity development workshop at Invimar in Colombia, also hopefully next year. So that was just a very quick overview of where we are with OBON and what we're planning. So there are some contact details and social media handles there if you'd like to get in touch or receive updates on what we're up to. So we will now move into the project presentations. And we're going to start with Filipe presenting from the University of Minho, presenting a couple of projects. I think the two projects that are that are kind of linked, both focusing on applications of DNA-based approaches for fisheries monitoring. So Filipe, if I can see your screen, so I think you're good to go.
I'm here. Yes, can you listen to me?
Yeah.
Yeah, okay. So hello, everybody. Good morning, good afternoon, good evening. So I'm going to present a work that is made by a large team, a large international team, which I will show you later on. And it's focused on the use of DNA-based approach for fisheries monitoring. This is coordinated by the University of Minho, where I have my research group. And the overall goal of this DNA decade action is to assist fisheries management with DNA technology. So, of course, environmental DNA can have a great potential and be very useful for monitoring adult fish fauna. But we are also very much interested in ichthyoplankton because ichthyoplankton, fish and eggs and larvae are extremely difficult to identify from a morphological approach, but they can provide precious information to assist fish stock assessment, like spawning seasons and areas, estimation of the recruitment and of the reproductive stock. So to start it, I will just present the team of this decade action. It's actually composed of two projects. One of them is called a fish DNA scan. The other one is called fish DNA monitor. These two projects are funded by the Foundation for Science and Technology in Portugal, and also the second project funded by the Hakai Development Network. And these projects involve Portugal, Cabo Verde, Brazil, Guinea-Bissau, and Spain. So except Spain, all of these are Portuguese-speaking countries, and that's one of the important features of this action. And of course, we are involved in OBON as well. So there are a number of different institutions that are involved in this project from the different countries. But I would highlight the governmental fisheries research agencies, which are IPMA from Portugal and the MAR, Instituto do Mar from Cabo Verde, and also INIPO from Guinea-Bissau. So when we started with this project, we had some technical challenges to face. So we wanted to do basically DNA metabarcoding, targeting marine fish in tropical Atlantic regions. So in order to be able to have a comprehensive monitoring of the fisheries in these regions, we wanted really fundamental to have species level identifications and attempt to minimize as possible the false negatives. So these are technical questions that we started to address. And among the challenges we had, the first questions we had was which marker regions should we use? Because there are different marker regions that have been applied for this purpose, and they have different strengths and weaknesses. Reference libraries are more or less complete depending on the marker, but they are particularly incomplete for tropical ichthyofauna. So we have to take this in consideration. Some markers have more discrimination power at the species level than other. Primer bias also occurs and no target amplification. So all of this we have to be taking into consideration. But another important question that we had at start was what volume of water should you filter to use for eDNA analysis? So here I start showing what we have done regarding analyzing the discrimination power of commonly used eDNA amplicons. This was a paper that was just published maybe 1 week ago. And so I will provide just a brief description of what was our conclusions about this topic. The target of this paper was to analyze the discrimination power of 9 genomic regions from 3 mitochondrial genes, 12S, 16S, and CO1, and especially the amplicons that are usually implied more frequently. And we used the mitochondrial genomes from over 700 fish species and from 10 fish orders to do this analysis. We did a second analysis just with the amplicon data directly from GenBank, but I'm not going to show it here. So the mitochondrial genome analysis showed us that there are differences, important differences in the discrimination power of different markers. We have them here listed according to their color, different markers and the different regions that were analyzed. And looking at the primers, the main used primers, we see that CO1 is the one that has the best resolution at species level, followed by 16S berry, and then MeFish on the 12S. And so there are some important differences here, but we also noted that it depended a lot on the fish orders that we are working on. Some fish orders, these markers may be much more proficient in discriminating species than other. And so overall, our take-home message is we need to keep this information in consideration when we do the eDNA-based monitoring, especially for fisheries applications. Then we also looked at different markers, different primers, and different water volumes. And for doing this, we made a partnership with the Lisbon Oceanário, which has a very big tank with 61 resident fish species. So it's particularly suited as a positive control for analyzing this type of parameters, analytical parameters that can affect the result. So we looked at the 5 different volumes of water from 0.5 to 10 liters and in triplicate these volumes. And for each of these triplicates, we have employed 3 genetic markers, CO1, 12S, and 16S. and also their respective primers. And we are showing here only 6 primers. We tested more, but these were the ones that function better. We show results for 3 different CO1 primers. 2 of them are cocktails that have already been published, and also the MeFish 12S and the very atoll 16S, Fish 16S primer. So when we look at the capability of detection of different markers and primers, we have here a summary of the percentage of the species detected when we put together all water volumes combined. And we can see that fish 16 is the best primer with 69% of the species. But this is still a low amount compared to what we would like to have. But in fact, some of the species in the aquarium tank did not have their sequences in the reference library. So if we look at in relative terms, compared to the available reference sequences for different markers, the percentage of success improves to 86% in 16S and 77% in 12S. Still, we are quite a bit below the 100% we should be the optimal finding. And regarding the volume of filtered water, you can see here by these graphs for the 5 of the primers that there were a very clear relationship between the volume of water and the cumulative number of species detected. So it really can make a difference on our detection ability, the volume of water we use for filtering. And with 5 liters of water, the maximum we could do was about 80% of the species. So this is something also that has to be taken in consideration during field sampling. Of course, we have to have here a trade-off between the sampling effort required by large amounts of water and the capture efficiency. So we came up with a 2-liter amount of volume of water as an intermediate optimum for us to work with. Then we went to the natural environment, and we looked at monitored using this kind of approaches, ichthyoplankton, and using at the same time morphology and DNA-based approaches. And we employed a multi-marker approach. We published this also in late August, so this is also very, very recent. But I'm going to show some data that we have not published yet, and where we applied this same approach. And this was a study that we recently did in that we still for publication. It was in looking at the seasonal variation of ichthyoplankton in an estuary in the southeast of Portugal, right next to the Spanish border. And what we did was that we made a monthly ichthyoplankton collections over a period of 13 months. And we did it on 3 sampling sites in the lower estuary and looked at morphology compared with DNA metabarcoding. So we had the backup of the morphology to back up our molecular data. And we used the 3 markers and 5 primers for this analysis. So when we look at the efficiency of the markers, what we can see is, first of all, we get overall 89 fish species during this 13-month period. And the 16S was the best primer set. It detected 65 species. But then if we add one more primer, each time we add one more primer to our analysis, we improve considerably the number of species detected. And we can see here in the opposite direction, we created a scenario here which we would analyze the data only with 1 primer set. And if this was the case, we would miss a considerable amount of the diversity that we find out at the end. So for instance, 16S was the best primer overall, but still we would have missed up to 27% of the fish species if we were using only 16S. So the combination of these different primers and markers really helped us to have a more comprehensive detection, and also to be able to minimize the false negatives in our study. Now, looking at this graph, it's very interesting because finally we start to have here some information that can be very useful for fisheries management. So these are the different species that were captured in the 4 different seasons. We distributed the data in 4 different seasons. And we can see here that some species, 18 species actually, occurred all year long, and some other only occurred in particular seasons of the year. So this is important for fisheries management. A good part of them were, as expected, were fish species that were reproducing and releasing their eggs and larvae during summer and during springtime. But there are also cases that only in autumn or only in winter, or different combinations of the seasons. So this information can be useful for fish stock management to know better the fish spawning areas and seasons. But there was more that we done. So this was a bit the scientific part, the main scientific results we had. There's more than this, and you can check in our website. But we also did a number of training and dissemination activities. And a key point was that all of these activities were running in Portuguese language. We did several workshops, 3 workshops in total for the fish DNA project, fish DNA scan project. And we also had some mobility of researchers. So we received the Minho other researchers from other Portuguese institutions, researchers from Brazil, researchers from Cabo Verde, so that we would harmonize procedures and provide some additional training all along the pipeline of DNA metabarcoding. We had a final event, which was very relevant. This event was in the island of São Vicente in Cabo Verde, and we had a number of activities there. We had several public seminars showing the results of the project and promoting the project. We also had a discussion panel with different experts about the use of DNA tools in fisheries research. And also we had meetings with local entities like the University, Atlantic University, Technical University of Cabo Verde Rectorate, with the Governmental Fisheries Institute, which is also part of our project, IMAR. We also met with them and we discussed methodologies with them. And overall, this had a very important impact also in the media in Cabo Verde. And we had news about the project and the workshop in the national TV, radio, and press. But we had also the other project I mentioned about, which was the fish DNA monitor. And we also had important activities here regarding training and capacity building. Fisheries is a very important sector in these countries, Guinea-Bissau, Cabo Verde, and they are in a position to use some of these new technologies to help them in fisheries management. So we did our inaugural workshop here at the University of Minho. We invited our colleagues from Guinea-Bissau and from Spain, but we also had last May a workshop at the Governmental Fisheries Institute of Guinea-Bissau, where we explained in broad terms the molecular methodologies to the staff of this Fisheries Institute. Also, we had opportunity to provide the first training to staff from this Fisheries Institute in the Guinea-Bissau. And luckily, we have got a fellowship for a new PhD student from Guinea-Bissau that is here and is going to be pursuing a PhD in a combination between Minho and Inipo. So this is going to be, we believe, a very strong way to transfer knowledge and capacity building to Guinea-Bissau. We also did some local sampling for this workshop to train the staff from Inipo, and we went to the fish markets, and we also collected eDNA in different locations, and we are now processing that data. In the future, opportunities will show the results from this second project. So the previous project I told you about already finished, and this one will continue at least until the end of 2025, we expect. We also had some dissemination events. We have public seminars that were, the audience were people from different research institutes in Bissau, not only from the Fisheries Institute, but biodiversity research institutes. And we had meetings with local entities like the Ministry of Fisheries. We had a great support from the Fisheries Minister. And currently they are developing some new facilities for this research institute, and we are helping them supporting the setup of a molecular lab so that they can, in the near future, start to using these approaches and implement them in their own country. We are also doing a lot of work in terms of reference library building for Guinea-Bissau, but we will show this in future opportunities. So basically, this was what we wanted to present you. Thank you very much for your attention. This was the work of this big team that you can see here from the two projects. And also we had the precious funding from FCT and Hakai network. And thank you also to OBON for this opportunity to show our work. Thank you.
Thank you very much, Filipe. That was so interesting. I forgot to watch the time, and I probably let you go over a little bit, but that was very interesting, both on the research side and on the sort of capacity building, and how you're now working to get the those results into the sort of policy applications. Are there any questions? I don't see any in the chat. If anyone wants to just put their hand up and ask a question, that's fine. If not, it would be great, Filipe, if you could, if you have the links to those publications that you presented, if you could stick those in the chat, so that people can access them directly. And I was thinking we could also put them on the OBON website, where we're trying
I'll do it.
to...
I'll do it in a minute. Okay.
Great. Yes. Thank you. I think we have a section on the OBON website where we want to share publications that, you know, like you have done, have kind of looked at the possible limitations of eDNA and you know, studies that have kind of looked a bit more into how quantitative they are, or how good they are at detecting different species. Okay, so if there are no questions, we will move on to Colleen from the Hakai Institute, who will be presenting the projects that she and Matt LeMay and others are working on over in Canada. Okay, I can see your screen.
Sorry, I just had to unmute. Slideshow. All right. Hopefully that looks okay. So thank you for this opportunity to share an update about the Hakai Institute Biomolecular Observing Network, or HIBON, as we call it. And in particular, I'll focus a bit on the community science component of our biodiversity monitoring network in British Columbia. So the Hakai Institute supports long-term ecological research on the coastal margin of British Columbia on Canada's west coast with 2 focal field sites, one Calvert Island here on the central coast of British Columbia, and then Quadra Island in the northern Salish Sea, about 100 nautical miles north of Vancouver, British Columbia. So HIBON has 3 main components. The first is our Ocean Biomolecular Time Series, where we bring together DNA-based biodiversity surveys and ocean observing data to uncover the drivers of community change on the smallest players of the marine food web. So here we have, while our focus for this project is predominantly on microbes, so here you can see some of our major microbial taxa that we have over time paired with some of the physical and chemical measurements that we make. We are also employing eDNA-based approaches to explore the biodiversity of higher trophic levels, including zooplankton, as part of this component. Another facet of the work that we do as part of HIBON is our efforts to expand biodiversity genetic databases. So we combine high quality photography and expert taxonomic ID, as well as DNA barcoding to contribute to the global genetic reference databases and inventory local marine biodiversity. To date, we've collected over 17,000 specimens, many of which we've collected on during BioBlitzes that we have hosted, with another 1,400 being collected as part of our Quadra BioBlitz, which we hosted earlier this year, which was a 2-week period where we were collecting samples. And approximately 20% of the specimens that we collect are new to genetic reference databases. So it's really helpful. This kind of feeds back to our then more eDNA-based approaches to survey biodiversity. The final component of HIBON is, and the one that I'll focus mostly on today, is our community science eDNA observatory. So the coastline of British Columbia stretches for more than 26,000 kilometers, which is pretty incredible. And so if we want to think about monitoring and inventorying the biodiversity in the region, how can we work at this scale? Like, as an institute that's kind of based in 2 main places, it's hard for us to do this alone. So our solution to this question was to employ an environmental DNA-based approaches to assess biodiversity, and then to reach out to coastal communities in British Columbia to see if they were interested in collecting eDNA samples to support this coast-wide network of biodiversity monitoring sites. And so from this came our Integrated Coastal Observatory, or ICO. The ICO program began in 2018 and draws on community partners to collect monthly eDNA samples. Our partners range from coastal First Nations to NGOs to other scientific institutes, individual citizen scientists, as well as the federal and provincial government groups. We employ accessible and low-cost methods for sample collection, which I'll show you some pictures of coming up. And while we originally thought we would focus more on marine invertebrates, since that's kind of a strength of the Hakai Institute, it quickly became evident that a lot of our partners were really interested in fish in their waters and maybe not sea stars. And so we pivoted and now target fish biodiversity using the 12S MyFish marker. Now I wonder if we should use 16S. To look at, to inventory fish biodiversity. And then we share our results through a publicly accessible website, which I'll share. I'll get into more of these details in all these coming slides. So what are the primary motivators for our partners to join ICO? Many of them are interested in basically in establishing baseline information about local fish biodiversity, as well as species at risk. They're wondering if we can use eDNA to look for a certain species at risk in the region. And then helping to understand the long-term patterns of biodiversity in their region. Some are also interested in early detection of invasive species. And then through this all, some partners have hopes of using the data to support conservation and stewardship efforts, to inform local management decisions and monitor biodiversity of marine protected areas, as well as Indigenous protected and conserved areas here in British Columbia. And so together we envision that ICO is a means to measure like ecosystem health and track biodiversity and track impacts of climate change and other disturbances on marine biodiversity in the region. So the ICO project is successful in large part due to the dedication of our program manager, Sue Velasquez. She's pictured right here after training a new group of people earlier this summer. Or I guess that was in the spring. She manages partner recruitment and relationship building, which is a really important part of maintaining a community science network, we've learned, as well as training and then also project organization. We currently have 17 active partners, collecting samples from over 25 sites across the region. And the sites that are sampled are selected by our partners with guidance from Sue if they need it. And we hope that by the partners picking sites that they're interested in, that will help them kind of ensure continued interest and dedication to the project and help them kind of target particular areas that they're interested in conserving or protecting. And then finally, we've created a training video to support partners in the event that Sue cannot make it out there to train them. This was particularly important during COVID when we couldn't go to train anybody or train anybody. So have a QR code if people want to watch that video. You can also, it's available at our ICO website as well, or that's the main place where you can find it. But yeah, this has really been important for training new partners, as well as kind of a refresher if someone is coming back onto the team and hasn't collected a sample in a while. Yeah. And so then our ICO eDNA sampling kits are pictured here. You can see that they're kind of based on sort of easy to access items or easy to buy items, though we provide them for our partners from like a garden sprayer pump to prime a pump. And then we send our partners fully cleaned water bottles and everything they need to collect and filter their samples and record their metadata. And we also provide a long pole sampler for sample collection, which we give to the partner at the start of training. And once our partners have filtered their samples and send them back, they send back the samples and the whole kits back to us. And then we have a whole lab at our facility on Quadra Island, which we call the decontamination lab or the decon lab, to clean all the water bottles and get everything ready to ship back out to our partners every month. So it's kind of just a revolving door of kits going in and out, trying to make things as easy as possible for partners to collect samples and maintain this time series that we've started. So here are a few pictures of some of our partners in the field. So it's a beautiful area to sample with very rich fish and invertebrate diversity. So we are definitely grateful for all that they are doing to help maintain this network. And then we also, a lot of our partners were kind of curious what happens to the samples after they leave their hands. And so in the past year or so, I think we partnered with our media team at the Hakai Institute to make a video basically explaining what happens to the samples after they leave the hands of an ICO partner. So that I think has been helpful because we're just very interested in making sure the partners kind of want to keep sampling and when they have questions, we try and address them as much as possible. And this is just a picture of our genome lab on Quadra Island. So another important part of our network is sharing data back to the partners. So our web and data visualization team has worked together with our ICO and science and our ICO science and tech team to create our ICO website. And I'm hoping I can bring it up here. Basically it's a pretty simple website where you can select, like grounded with an interactive map where a partner could like select their site, Quadra Island, I'll pick that one, and then see all the types of fish that are available or that we've observed. They can stretch out basically how long the time series is, or shorten if they want to focus just on a particular year. And then look across, or look across maybe all the sites and see kind of where do we find certain groups of fish. Hopefully, so go back to presenting. Yes. Yeah. So then another important part, and I guess like another thing that we have been also interested in is our partners have expressed interest a lot in wondering whether or not the samples that they're collecting can be used, oops, go back, for looking for invasive species in the region. A lot of people are really concerned about European green crab, which is an invasive species in British Columbia that destroys eelgrass beds, which are important habitat for herring and juvenile salmon and other organisms in the region. And so we decided to see if we could use our ICO samples, which are collected from the surface, I didn't mention that, and see if we could implement a ddPCR assay to assist in potentially early detection of these using this big kind of sampling network that we have. So here, basically what we've done is start by looking at like basically picking two sites, which are highlighted here. Oops, my mouse on the wrong screen, sorry. Two sites that are highlighted in pink where we know green crab adult populations have been established, and then seeing how we can detect the European green crab using the validated assay that exists in the literature. So basically, if we go back, first, it appears that it's pretty low levels of detection, but we are able to detect green crab. It's kind of driven by outliers. So if we zoom in and get rid of the kind of really high outliers, we can see that at these established sites, we're detecting green crab all year long. We had kind of hoped that maybe by kind of looking at these established sites, we could pinpoint a time of year when we could say, like in June, we're going to test all of our ICO samples to see if we're detecting European green crab at sites where it's known to be present, as well as sites where it's not known to be present. But it seems like, especially at sites where it makes sense, where there's established populations, we're detecting European green crab most of the year. So now we're going through and testing, basically focusing on spring, summer months and looking at European green crab across this whole range of sites pictured here. And so hopefully this is something that we are hoping to kind of respond to our partner concerns and incorporate these data into our ICO website by hopefully the end of this year. So yeah, looking ahead to 2025, that is one of our goals, is to get the European green crab aspect of the invasive species monitoring aspect of ICO up on our ICO website. Another thing that definitely one of the challenges in having a community science program is kind of maintaining interest in the program among the partners. So we're really keen to, this year we'll be hosting our first gathering of all of our ICO partners, which is a pretty long time coming since we established the network in late 2018. But we're hoping that this will help the partners have a chance to kind of share with us any challenges that they're facing, for us to share back with them like all the amazing work that they've contributed to. And then also, help renew enthusiasm for ICO, and then maybe help us make some more connections and recruit more partners. Another big aspect of what we are going to be working on in the next year or so is formalizing our data management plans and the data sharing strategy for the ICO network. Right now, it's just pretty informal with our partners, but if we want to be able to share this data more broadly beyond the website visualizations, which are kind of not the raw data, obviously they're not sharing the raw data with the partners. We want to be able to kind of see to what extent we can make this data available publicly. And so I guess one of the things we were asked to talk about today was thinking about how OBON can help support the work that we do. And I think it could be really interesting to have some discussions about not only the FAIR principles of data, but also the CARE principles. We work with a lot of Indigenous groups, and it's really important for us to do this part of our project correctly, I would say. Yeah, and so those are the main parts that I wanted to update people on lately and share about our ICO project. This work is done through a great team of people on the Hakai side, from our program manager to our lab manager, as well as many lab technicians, bioinformatician, and it's really critical to have the website, I think, and the data visualization aspect of the project. And then here's just some of the logos for some of our partners, though not all the individual partners have a logo for their family. Yeah, so our work is really built on the kind of this partnership between Hakai and a large group of people to really see if we can start to kind of map biodiversity on the British Columbia coast. And we couldn't do this alone at Hakai, so it's been really important to cultivate this network of external partners as well. Thanks.
Great, thanks very much, Colleen. That was fascinating, and always great to see how well you're doing citizen science, the eDNA with citizen science. I know a lot of partners in OBON are always interested to hear about how you do it, and what lessons they can learn. I don't know if there are any questions. I don't see any hand. I do see a hand. Lika.
Hi, Colleen. Thank you. Great presentation. I'm just curious, what was the first impact on when you approach communities? How did they receive this, this idea of such an innovative and, modern technique, and how they could help with it?
Yeah, I mean, I think most people were pretty, I think environmental DNA was becoming kind of public in the, or becoming visible in the public's eye around the same time that we proposed this to partners. And a lot of people actually were coming to us because sometimes we're in communities collecting samples. And so they'd be like, well, can we be part of this or help out with the research that you're doing, or be part of the research that you're doing? And so this seemed like a relatively easy way to engage people. It doesn't require any sort of overly fancy sampling or sample preservation or anything. And so at the start, I mean, and a lot of it has really become like people coming to us wondering if they could be a part of the network, or and then once they understood that what environmental DNA could tell them about their environment, they were kind of keen to go on. But it is definitely an effort to get out there every month and collect samples. And currently the method we use with the priming a garden hose or a garden sprayer pump, it's not like a speedy process as the partners filter the samples. And so one of the things that we're looking into this year is to help see how we can streamline and kind of maybe speed up that process by building some little DIY peristaltic pumps to see if that might help speed things up, because I know sometimes people are not super keen on the slow process there. So this is, kind of maintaining that aspect of things is definitely.
And do you notice a specific
samples in the
a profile on your citizen scientists? Are they usually young or older, more of traditional communities, or more urban?
It's definitely.
Have we lost?
Are kind of.
Oh, I thought it was just me.
Oh, sorry. Can you hear me now?
Yeah.
Yes.
Okay. Well, at least it lasted during the talk. It's a range. The kind of individuals are often older, I would say, but when we're working with First Nations, we're working a lot with what are called their guardian watchman groups. And those people, or those groups, span a range of ages and experiences. And it's part of a bigger network in British Columbia to help First Nations be stewards of their local environment. So that is definitely a big age range and experience range. So
amazing. Thank you so much. Thanks.
Okay, thanks very much. So now we're going to move on to Alice Ortmann, also from Canada. Changing topic slightly to the microbiome. So Alice, if you're ready to go.
Okay, can you see the slides?
Yes.
Yes. Okay.
Perfect.
So yeah, this is going to be a very different kind of project than the previous two projects, in that there is no data collection and sample collection and processing. So this is the Observing and Promoting Atlantic Microbiomes, which is our Ocean Decade version of the Marine Microbiome Working Group. And this working group is a collaboration of Canada, the US, and the EU. So I am the Canadian co-chair, Kelly Goodwin from NOAA is the US co-chair, and Stéphane Passant is our EU co-chair. And see if this works. So the main goals of the Marine Microbiome Working Group are really to promote marine microbiome research. And this was a recognition that microbes tend to get overlooked in a lot of the observing networks, or just in marine science in general, in biogeochemical modeling, or in physical modeling. We tend not to think about the microbiome very much. And so we wanted to promote marine microbiome research within the scientific community, as well as within kind of a general public kind of outreach activities. We also wanted to develop a strong collaborative international network of marine microbiome researchers. So bringing together people who are all working on these little critters across, initially it was the North Atlantic, and as we've been expanding it, it's covering the all Atlantic now. We also wanted to integrate new technologies in collaboration with industry, and initiate and support policies to foster sustainable marine development. And one of the major accomplishments that we have made in the past is developing the AORIA Marine Microbiome Roadmap. And so this roadmap outlines some different pillars of focus, including environment and climate, food security, and biodiscovery, as well as cross-cutting initiatives such as outreach and data management. And so we've been working to move past our roadmap and hopefully implement our roadmap. One of the things we wanted to do was reach out and expand our network. So as I said, we were initially the EU, Canada, and the US, but we've also been collaborating a little bit with the AA Biotech Joint Action. So this is a biotechnology focused group, which has a strong emphasis on marine microbiomes as well, that's led by Brazil and South Africa. And this is a group that came out of the Belém Agreement, which was between the EU, South Africa, and Brazil. So kind of collaborating with these guys, we've changed our name a little bit. So we're now calling ourselves the AORIA BAM Group, or the Blue Biotechnology and Marine Microbiome Forum. So that's our website name, with an emphasis on the fact that microbiomes really underpin a lot of the biotechnology, and specifically blue biotechnology, that people are looking at the oceans to support. We've also been reaching out to researchers outside of these official agreements. So one of our recent newsletters actually highlighted the work of Agatha Alkemitsi from Nigeria. I actually met her when we were the Microbiome Working Group was actually hosting a session at last year's American Society of Microbiology meeting, and Agatha was presenting her results. She's been working on oil spill mitigation in the Niger Delta, and reading her story, it really highlights how science can be very different for people when they're living in a place like Nigeria, where some of the stories that she was sharing are, it's dangerous to go collect samples and do some of the science. And so it's a very different experience that we were sharing there. One of the things that we're working to do is sort of find our way under this new AORIA agreement. So the All-Atlantic Declaration was signed in 2022 and expands the previous agreements that were sort of north and south, separating the Atlantic, into this all-Atlantic focus. So originally it was Canada, the US, EU, Brazil, South Africa, Morocco, and Cabo Verde, and Argentina. Since 2022, Norway and Iceland have joined, as well as the UK, and Senegal was actually announced as the most recent partner at a meeting just last week. And the IOSC and UNESCO is now listed as an official sponsor of AORIA. So, as I said, there was a meeting last week in Ottawa, and this was the most recent of the All-Atlantic Ocean Research and Innovation Alliance forums. at the forum, we were discussing the 2 areas of action that have been identified for AORIA. The first is ocean and climate for coastal resilience, and the second is ocean observing and modeling. And so as the Marine Microbiome Working Group, or combined with our BioTechMar partners as BAM, we're looking for how we might play a role in these 2 action areas. One of the areas that makes the most sense would be focusing in working towards supporting the ocean observation and modeling area of action. However, it was really interesting at the meeting that there was very little biology discussed at all in the meeting. In fact, I think eDNA was mentioned maybe once, and microbiomes were mentioned once. So there's really a strong focus right now in this group on the physical chemical observations as opposed to biology observations. There's also the potential to host an ECOP through the @Sea network. I have a microbiome-focused mission that will hopefully occur next year. And so we'll be looking to see if we can find a microbiome researcher who could participate in that mission through this @Sea network. One of the other ways we've been reaching out is through scientific publications. So over the past, I guess since about October of last year, we've had 4 different publications come out. One was in Frontiers of Microbiology. This was with Kelly Goodwin and Anaïs Lacourcier. And this was their editorial that was part of the special edition focused on microbiomes and best practices methods. We also had a policy and practices review published in Frontiers of Marine Science, as well as a couple of other publications that were really focused on how marine microbiomes can fit into a blue bioeconomy, how they could be used to support sustainable fisheries, or bioprospecting to support discovery of whether industrial applications or new food products or something, or drugs. We've also been continuing to work towards our sparking fascination with microbes through existing and new visuals. So the QR codes here, the one would get you linked up to our activity and puzzle coloring book that is focused on the microbiome. And we had originally 3 little videos that we have now actually expanded. And so we now have 8 new short videos. And I don't know if I'm going to be able to make this work, but this is just one of our short videos on pollution.
The marine microbiome is being used to break down petroleum that has spilled into the ocean. Floating plastic waste in the world's oceans is first colonized by the marine microbiome. Perhaps the microbiome will be a key to mitigating ocean pollution. Discover more.
So that was just a short example of one of our videos. We keep them very short so that they're very good for sharing on social media, and they cover a variety of topics, with the videos mostly coming from NOAA's library of videos. The other thing we've been working on is increasing our social media presence. So we hired a new social media manager in 2024, which has increased the amount of postings that we have on all of our different social accounts. And so we're very excited that this increased our engagement rates between 46 to 1700%. But the reality is those numbers, that if you go from really, really bad, 1700% increase in your engagement just kind of gets you to better. So it's a very slow process. And one of the things that we did want to mention is that feel free to reach out to us with any of updates on your projects, even if they're not microbiome associated. We do share a lot of information about eDNA and genomics, as well as the microbiome work. So we'd happy to either share your information, setting up our own posts, or link to any posts that you have. And also, if you can like and share our posts, then that, of course, will help build our network. And so we, of course, would be happy to like and share all of your posts too, if you've got social media that you'd like us to emphasize. So moving forward, we've submitted some new ideas for some new activities that we're reaching out for. Like many people, we need funding to make these work. One of the things we would like to do is develop some bidirectional exchanges on some early career microbiome researchers so that we can really develop a much stronger network. And so here what we'd like to do is have researchers go from say a lab in Europe down to a lab in South Africa, and then somebody from South Africa go back up to that lab in EU, so that we're getting people moving in both directions, so we get a better understanding of the different situations and capabilities that all the groups have. We're also reaching out to the Oceano CSA, which is the AORIA Coordination and Support Action, to understand how we potentially could fit into this ocean observing and modeling action. We're also working on developing some new partnerships. So there's the World Microbiome Partnership, is a new initiative that's looking to draw all microbiome researchers from all sort of realms wherever microbiomes are, so whether it's human health or environment or agriculture, and kind of make this giant network. And they're having an inaugural summit in December this year in Paris, and both myself and Stefan will be presenting during the summit. And we've also been spending some time this year kind of for reflection about where we are and where we'd like to move forward, and really working towards developing a new plan in 2025. And so with that, I'm just going to share a million QR codes with all of our socials. So feel free to reach out to us on those, or reach out to me personally, and I can connect you with our social media manager. And with that, thank you.
Thank you very much. A lot of very useful information there. I think our communications officer, Fiona, is on this call, so she will probably be making a note to amplify anything that you post on your social media. And then on the ECOPS and shipboard training with the Sea Network that you mentioned, Pogo is involved. So Lillian Krug, our scientific coordinator, is on, I think, their advisory board. So we have funding for shipboard training, and we also try to provide or share opportunities with the Sea Network. We actually hopefully we'll have a shipboard trainee on the next AMT, the Atlantic Meridional Transect cruise, which is happening next month. So that will be linked up with the AMT Omics, which is also an OBON project. So a lot of possible collaboration there.
Yeah, no, the @Sea Network looked like an excellent opportunity. And so that's definitely something I think that many of our participants might have opportunities that we could offer.
Yeah, excellent. Are there any questions on this project? Time is running on, so we have one more presentation which is pre-recorded. So I think if there aren't any questions, or if anyone comes up with any questions, please feel free to put them in the chat for any of the speakers who I think are still around. So thanks, Alice. And Luz, if you could share the presentation from Saara Suominen, who will be, I think, talking about 2 IOD-led projects, which are both global projects using eDNA. One is on focusing on invasive species, and the other one is looking at marine World Heritage sites. Okay.
Hello, everyone. My name is Saara Suominen, and I am working for the Ocean Biodiversity Information System, which is part of the Intergovernmental Oceanographic Commission of UNESCO. I'm here today to talk to you about 2 OBON endorsed projects, which is the PacMAN project and eDNA Expeditions project. I will first give you just one slide introduction into what is OBIS in case you're not aware. Then we'll talk a little bit about the 2 different projects and especially what we have been working on this year. And finally, just one slide on what eDNA data in OBIS is and what our future plans are for that. So what OBIS is more than a database. We are a community of practice of data managers, scientists, and users. We get data from over 1,000 different data providing institutions. We are a gateway to marine biodiversity data and information. Our priority areas are, of course, in data mobilization, so bringing all that data together in one place so that it is searchable and findable and accessible and comparable, but also in data application, so in the use of this data that is in our database to provide help for decision-making and fit-for-purpose data products and packages. So that's in a nutshell of what OBIS is, and I'm already going to go into what our projects are that are part of OBON. The first one is PacMAN, which is a marine invasive species project utilizing eDNA for a monitoring program. PacMAN stands for Pacific Islands Marine Bioinvasions Alert Network, and this project is implemented in Fiji. This project had several clear pillars that it was built on to kind of try to make an operational monitoring system that would actually bring value to the local community. And the first one was really that we worked closely with stakeholders to identify their needs. We developed the monitoring program together with an international scientific advisory board. We developed the full complete data workflow from raw data to the decision support tool. And of course, we had open access training to provide capacity development and hopefully continuation of the project and the capacity in Fiji around this. So first of all, we started with a needs assessment of the local community, and it was really clear from the very beginning that there was very little information on marine invasive species available in Fiji and at the harbor, so we were mostly working in the Suva harbor. So we decided together with the international scientific advisory boards that the monitoring plan includes also baseline analyses of the biodiversity at these sites, these hotspot sites. And so that meant that what we did was collect 3 different types of samples, which was settlement plates, water samples, and plankton net samples at 4 different sites at the Suva harbor. And these were then analyzed for community composition with metabarcoding with 2 broad range biomarkers, which was the CO1 and 18S. In addition, we did develop also a few qPCR assays because one of our objectives was to also have that early detection rapid response, which is really only possible with quantitative PCR. So at least we wanted to get the community started with developing these assays and these methods. And all of this field work was implemented by our partner at the University of South Pacific, who developed the actual field workflows and implemented them. And our work at OBIS really focuses, of course, on the standardized data management and the data workflow. So from the PacMAN monitoring campaigns, we record the field data and the specimen data to Bluetooth data management platform, where it is easily accessible through APIs. The raw metabarcoding data is submitted to NCBI and is analyzed through our own bioinformatics pipeline directly into Darwin Core archives. So this is the data format that OBIS uses in our database. This means that the final formatting step is integrated already into the analysis. This data then flows into OBIS, and it's actually taken from OBIS into the decision support tool. So this means that we're not only looking at the data from a specific study, but we are looking at all the occurrence data that is available for our location of interest, feeding that into our decision support tool, and looking into the possible risk species to help the decision makers to identify what possible species can be an issue to them. The risk analysis is based on the likelihood of an invasion and the probable severity of the expected impacts. So we are looking at known invasive species, but also species that are not from the area, but are from similar thermal ranges that could possibly become an issue to the area. As you can see here, all of our workflows are open access. So the PacMAN bioinformatics pipeline is on GitHub and can be easily accessed and used from there. And in addition, all of our workflows for the decision support tool will also become available. Now, just to show you a short look at what this will actually look like. So this is really the newest version of the decision support tool that I got from our data manager, Peter Provost, yesterday. And you can see, for example, the risk analysis for the tunicate Didymnum perlucidum in Fiji. And this map clearly shows where the species is known and is known as invasive. So we can see that in Western Australia, it is considered invasive. And now we have our first detection in the Suva Harbor in Fiji. And it's something that we would highlight in this decision support tool so that the local managers can go and investigate further what the situation is in the Suva Harbor. We had multiple other possible invasive species detections as well from the eDNA. And here we have not been very selective. So we list many potential species, but we do indicate that the confidence has been low. For example, the risk is possibly low, and we indicate that there is a status that you should still review it, right? So here you can filter through what you're interested in, and of course, probably look at the very high risk ones first and evaluate if you need to investigate this further. So we really try to showcase this as the first line of information, but not be too selective in what we add here, because it's important for you to get that information of any possible invasive species if they potentially are a high risk for the environment that you are trying to manage. And of course, this PacMAN decision support tool is intended to be able to be used in many different areas of the globe. So it's not only linked to Fiji and the program that we have there, but as I mentioned, the data is taken from OBIS, so this can actually be used in many different locations and can be adopted. And we have the pillars that are needed to build such a monitoring program available. So we have a nice experience here of building a program together with the local community that I think might be interesting for other areas as well. And then on to our next project, which was the eDNA Expeditions project. This is a global citizen science eDNA sampling campaign. And so here we developed a very simple sampling kit, which was just a syringe and a filter. And we sent 20 of these around the world. So 20 to each location. And the local communities themselves developed a sampling day together with either school children or other local communities to go out and take samples from their Marine World Heritage sites. And you can see here some pictures of what this looked like in different parts of the world. And there are also more pictures and stories from these different sampling campaigns on our websites. So so far, we have done 22 Marine World Heritage sites. The sampling was conducted in, excuse me, 2022 and 2023. We shipped in total 550 eDNA kits to these sites, which I think we got back 400 that we were able to sequence in the end. So there were, in fact, a few sites where the political situation either didn't allow sampling or sending the kits back. So that's the reason why we have a few less than what we sent. And training was done really just by video calls. We had also a training booklet and different training videos to enable this sampling. And we engaged over 200 citizen scientists in this work. And what we have been now doing is really trying to find a way to showcase the information that we got from these sampling campaigns for the citizens and also possibly for scientists or for the managers at their local site. And we wanted to find ways to have an engaging dashboard where you could easily find the main information that we can give from this, These sampling campaigns were small. You had only 20 samples in often very geographically large sites, which had multiple habitats. So these are really just a snapshot that we could get from these small campaigns. So we wanted to rather showcase the diversity that we did get rather than saying something about the full diversity at the sites, because of course this we cannot do. We did also compare to OBIS and GBIF information from the sites, and we got around 10% of the total species known from most of the sites, which is quite good for one sampling campaign, but of course is not a full picture of the biodiversity in the site. But in this page, for example, you can see the results from one of our most successful sites, which was in the Aldabra Atoll in Seychelles. You can see that we got from those 20 samples almost 850 species, of which almost 400 were different fish species. One of our main focuses in this dashboard was really to make that eDNA data a little bit more concrete, to try to showcase what this means in terms of the immense biodiversity that we can actually detect. And so we decided to have a gallery of all the species that were detected. So this gallery will allow the citizens to browse through what species were detected in their location. And by clicking on an image, you will also get information, a small information on that species, as well as the actual DNA sequences that were found. So linking that a little bit to what the data is behind all this information. And hopefully this will also allow scientists to then confirm if how reliable this species detection is. We did do a lot of work in confirming species detections and reviewing species detections with local communities, but of course in such a global work, there may still be some misidentifications, but we hope that by giving all the information, people will be able to evaluate that for themselves. We also have a bit more scientific information. So for example, we will show the taxonomic composition across samples, where you can see, for example, the remaining unknown sequences in each sample. So the proportion of reads that still remain unknown, which I think is very crucial information if you are working with eDNA. We also show the differences between the different locations with alpha diversity and beta diversity measures. In addition, on other tabs, we have just a species list as a table, as well as chronoplots, so you can discover that taxonomic diversity and explore that. We also have a samples tab where we show all the different samples that were taken and the information on them. And finally, which I think is very interesting for many people and for hopefully the local managers as well, is my colleague Silas has done a climate analysis on these species that we find. So he's looked at the thermal ranges of the species that we detected and evaluated how close they are to the edge of their thermal range and what will happen with different possible climate scenarios. So which species will be at risk with different temperature increases at the site. So in this case, we hope that the managers can explore these tables, look into, for example, the fish species that they are interested in, see which of these species might be most at risk in the future when temperatures rise. So that's it on the projects and their developments. We have, and now I'll talk a little bit about what OBIS is doing in terms of eDNA. We really think of OBIS as a possible AS3 repository for genetic data. We have the infrastructure that is needed for collecting and sharing that data in a fair way. In addition, we have many guides on how you can add data to OBIS, and with our manual and different videos and training manuals. We are also developing data products, as you have seen in this presentation, that we hope will help decision makers use that eDNA data to their benefit. In addition, we are involved in many European projects for eDNA data standardization. For example, to mention one is the Marco Polo project, and we had a data analysis challenge where we are looking into using one standard data sets with many different bioinformatics pipelines, and we're sourcing that from the community and looking at how the outputs of those different bioinformatics pipelines differs to evaluate how robust the analysis methods are that the community is currently using for eDNA. So thank you very much. I'm sorry I couldn't be there in person, but feel free to send any questions that you may have to my email address, which is right here. So thank you for your time.
Okay, so thank you very much for joining. And yes, sorry about the technical issue there. And hopefully see you next week. Okay.
Thank you very
you. Bye.
much. Bye-bye. Thank you.